Hi, everyone. We thought we would do another short, whatever you call it, little Zoom webinar, I guess. That would be a good name. And I thought I would just talk for maybe a half an hour or so and just give people an update. We also have a Weston Price video conference that we're going to record today. So Sally and I are going to be taking questions. So this may come out after that, but you still should be able to access that. So again, thanks for joining me. Hope everybody is doing well. And I've obviously gotten a lot of questions around the book. And there's also been some new information that's come out. And I thought I would spend today just going over some of the basics of the things that people are asking me about, and then talking about four of this. Some of them are new studies and some of them are studies we didn't know about. And then at the end, I could give a little bit of a summary of just a way to think about health and disease that hopefully you can put in practice and use in your own life. All right. So a lot of people, one of the main questions I'm getting from not only lay people, regular people, and scientists, sometimes doctors, is how can I possibly say that this virus, the coronavirus, otherwise known as SARS-CoV-2 virus, which is reportedly the cause of the disease called COVID-19, how can I possibly say it hasn't been isolated, purified, and characterized, and then shown to cause disease when there's literally scores, if not maybe even 100 papers in the medical literature that have something in the title called the isolation and purification, or the demonstration that it causes disease, or something like that. So why is my opinion so much different than the conventional medical and virology opinion? Again, a lot of people have sent me those, and we actually, as anybody who's read the book knows, that we actually went through many of these studies, actually the 6 most important studies, many of them with the title of the isolation or purification of the new or the novel SARS-CoV-2 virus. And we went over them in the book and showed that, in fact, they neither isolated nor purified nor showed that this virus causes any disease in animals or people. But apparently that's still not, it's still not enough. So let me try this from a different point of view, because really what we're talking about is basic science, basic, not even just science, basic way that human beings think. That's really what I'm talking about. So let me give another example that hopefully will really drive this home. So let's say you're a person with and you're interested in studying something and seeing if it's true. That's what really we're talking about. That should be the basis of science. So the particular thing we're talking about, or hypothesizing, that means we're talking about a guess that we're hoping to prove, is and I want you to pay very close attention to the wording I use. The caffeine found in coffee beans, when taken orally by human beings, causes a rise in their blood pressure. So let me say that again. Our theory, or our hypothesis, is the caffeine found in coffee beans, when taken orally by humans, raises their blood pressure. Now, we all think that there's caffeine in coffee beans, and that may or may not be true. But let's find out. And let's find out whether that theory or hypothesis is correct. So I'm going to ask a series of questions. Unfortunately, I can't see a raise of hands to see who would answer yes or no, but I'll guess. So the first thing I do is take a bunch of coffee beans, and I have 10 people swallow the coffee beans. And some of them, let's say 8 of them, have a rise in their blood pressure. So the question is, does that prove the theory? And I would love to see a raise of hands, because I think nobody would say that proves the theory. And probably in that case, nobody would be dumb enough to actually swallow a bunch of coffee beans, because I don't think that's so good for you. But the reason is because there's obviously a lot of things in a coffee bean. There's oils and caffeic acid, and I didn't look it up, but my guess is there's literally scores, if not hundreds of other substances. So if our goal is to prove that it's the caffeine, you can't just swallow coffee beans, because that doesn't prove it's the caffeine. So, okay, let's do the next step. Let's take the coffee beans and grind them up in a coffee grinder. And then we put, because that's easier than swallowing beans, and we put those ground coffee beans in capsules. And again, we have 10 different people swallow the capsules. 7 of them raise their blood pressure. Does that prove it's the caffeine in the coffee beans? And obviously, again, the answer is no, for the same reason. So let's go a little deeper. So we take those coffee beans, and I happen not to be a coffee drinker, but I think probably some of you are familiar with the fact that you put it through some sort of a drip filter, and then you get a liquid called coffee, and the grounds, which is the sort of, cellular part, so to speak, that stays in the filter. So then you discard the coffee grounds, and now you have just liquid coffee. And then you feed that orally to 10 people and see what happens to their blood pressure. And again, 7 of them, their blood pressure goes up. But I think most of you would then agree that that still doesn't mean it's the caffeine, because there's probably many, many other substances, oils, aromatic things, caffeic acid, that's the only one I know, also found. So you don't know whether it's the caffeine. So the next step would be to put it in some sort of a chromatography apparatus, or more commonly, put it into a centrifuge with a gradient. And what happens in a centrifuge is it spins real fast for a certain amount of time, and everything with a certain weight creates a band in the gel, and that is pure whatever it is that weighs that specific amount. And so you may have 20 different bands separate out, and each of those is a specific chemical. And there's other ways of doing that, but that's the most common. Now, then you can take a very small micro syringe and suck that band out of the gel, and then you have to verify that you only have caffeine. And that's because in science, we're taught never believe anybody, for good reason. You know, you may not have spun it long enough, so you may get some other things in there. In other words, you have to document with some very clear evidence that in that band, you did it properly, and the only chemical you have in that band is pure caffeine. Now, if you can do that, and there's very clear ways of doing that, then if you feed that caffeine to 10 human beings, and all of them, or 7 of them, end up with a rise in their blood pressure, you have essentially demonstrated that it's the caffeine in the coffee beans that causes an elevation in blood pressure when taken orally. And that is how science is done, or at least that's how science should be done. You cannot say that it's the caffeine in the beans unless you've done those steps. And that's called the isolation and purification. And I dare say that I don't think there's any rational person or scientist who would disagree with that. The problem comes, let's say you do these steps, you grind it, you filter it, you centrifuge it, and then there's no caffeine in there. I mean, I think there's caffeine in there, But I would admit that unless we do those steps and demonstrate that there in fact is caffeine in there, I'm not sure there is. Once we do those steps, then I'm 100% sure there is. And if we do those steps and find that there's no caffeine in that centrifuge, either something happened to degrade it, or, which would be interesting and possible, I guess, except if the other chemicals come out intact, then it's not likely that that's the explanation. So if I get other chemicals but no caffeine, it's hard for me to explain why caffeine is the only chemical that degraded. So the general explanation, if I don't find caffeine in the centrifuge in a band, is simply because it's not there. So with that background, let me go over some of the studies that we have either found or were published since the book. And maybe some of you may have heard these, but let me just go over them. So I'm going to basically do this by citing the paper, and then I'm going to read a quote from the paper, and then I'll explain why that's important. So the first one is a paper published in a journal called European Surveillance, January 2020. The author is a guy named Corman and others. And the title is Detection of a 2019 Novel Coronavirus by Real-Time RT-PCR. So this is a paper in a peer-reviewed European journal about detecting this coronavirus. So here's a quote. Quote, the ongoing outbreak of the recently emerged novel coronavirus, 2019 nCoV-2, poses a challenge to public health laboratories as virus isolates are unavailable. So what does that mean? It means that it turns out these virologists, I think there was about 20 of them, were tasked with the job of developing a test for finding this coronavirus. But as they said, this is a challenge, and I would admit that that is one part of that sentence I actually agree with, because it's a challenge because they don't have any isolates of the virus available. In other words, they don't have the caffeine. They don't have an example of the virus. And it would be very challenging to make a test to find something which you've never seen. So I would agree with them on that point. The analogy I made, it's like you're told to look for unicorns, and you can look for hoofs and tails and hairs and things, but you're not allowed to actually see a unicorn. And it could be because unicorns don't exist. So then they go on in the study and say, quote, we aim to develop and deploy robust diagnostic methodology for use in public health laboratory settings, without having virus material available. And as I said, I had to read that a number of times because their job, what they're saying, is to develop a test that the test is based on finding a unique piece of this virus, knowing that this unique sequence could have only come from that virus. Therefore, they can look for the sequence instead of the virus. Yet, as they admit, and this is January 2020, they don't have any virus material available. And I can imagine how challenging that would be. So I'll leave that one for a minute. The next one, people have heard me say this. This is from a more recent study on the CDC document. So this is published by the CDC itself. The title is called novel coronavirus 2019 nCoV-2 real-time RT-PCR diagnostic panel published July 13th, 2020. On page 39 of this long document, we read the following, and I'll quote, "Since no quantified virus isolates of the 2019 nCoV-2 virus are currently available, assays, parentheses, diagnostic tests, designed for detection of the 2019 nCoV RNA were tested with characterized stocks of in vitro transcribed full-length RNA." Again, in other words, this is a document describing in July 2020, so this is well into this pandemic, of how they go about developing these PCR tests. And they say, quote, "There are no virus isolates available." In other words, they've never actually found the virus. They've never found the caffeine. They've never gone through those steps. Therefore, you don't know whether there's even caffeine in there. And so instead, they use their testing sequences that are based on stocks of in vitro, that means laboratory created, transcribed full-length RNA. In other words, back someday, they made these full-length sequences in the Petri dish, not from isolating them from anybody or anything. And that's what they're using as the blueprint, the template, to see whether they actually can find the virus. So that's crazy. The third is a paper that was published in June of 2020, also by the CDC, in CDC Bulletin Volume 26, number 6. The author of the paper is Jay Harcourt and others, and the title is Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease. And let me read you the quote from one of the sections in this. The authors state, quote, "Therefore, we examined the capacity of SARS-CoV-2 to infect and replicate in several common primate and human cell lines, including human adenocarcinoma cells, parentheses A549, human liver cells, parentheses HUH 7.0, and human embryonic kidney cells, parentheses HEK-293T. Each cell line was inoculated at high multiplicity of infection and examined 24 hours post-infection. No CPE was observed in any of the cell lines except in vero cells." Now, that's a big, long mouthful, and I don't expect anybody to understand what they mean or see the incredible significance of that paragraph. So let me dissect that a little bit. First of all, CPE means cytopathic effect. So here's why that is significant. So if a virologist wants to prove that a certain virus, isolated or not, causes disease, they have 3 different possible hosts in order to test it on. The first host obviously would be humans, people. The second host would be animals. And the third host would be tissue culture. In other words, tissue taken from a kidney tissue of a monkey, or human tissue from the lungs, or human fetal tissue, or eggs, or whatever other living tissue, because obviously we all know viruses, quote, don't grow in petri dishes or on non-living medium. So the question is, have we proven, because that's the point of this, is to demonstrate that this coronavirus causes disease. So there are obviously no examples where they've tested this isolated or not coronavirus on humans, because clearly that would be unethical, right? So we have no examples of this. So we have no direct knowledge that this coronavirus causes disease in people. So the reason for that, they say, is because that would be unethical. Now, what about with animals? So as we know, and we dissected in the book, there's essentially two studies that have attempted to use unpurified, unisolated virus fluid. That would be like the coffee. So they injected the coffee, which has a lot of things, into a bunch of wild mice, and none got sick. And then they injected them into genetically engineered mice to have fur problems when stressed, and a statistically insignificant number of them, in fact, got fur problems when injected with this unpurified stuff, which supposedly contained the virus in it. And so essentially they demonstrated that they couldn't make normal animals sick with the virus or a fluid supposedly containing the virus. And even animals, i.e. mice, programmed to get sick, they could only create a statistically insignificant number of them got sick in a way that had nothing to do with what's called COVID-19. In other words, they have no evidence that this virus or this fluid containing the virus causes disease in animals. So the next step is you have to do it on tissue culture. And in fact, most virological studies are not done on humans for supposedly ethical reasons. They're actually even anymore not done on animals because they say there's, quote, no animal models for this disease, which is an interesting thing to say. What the translation is that it doesn't make animals sick. So most of the proof that the virus causes disease, or more technically, the fluid which supposedly has the virus in it, is by inoculating this fluid containing the virus onto various tissue cultures from various sources. And so that's what they did. And shockingly, what they report, and by the way, we all know that if you just inoculate virus on tissue culture, nothing happens. We know that for 50 years. So what they have to do in order to make the, quote, virus have a cytopathic effect is first starve the tissue. They do that with what's called minimal nutrient medium, which means they take away the nutrients from the tissue. And then they add usually gentamicin and amphotericin, which are two known nephrotoxic drugs, to a tissue culture of kidney cells. Nephrotoxic means toxic to the kidney. And of course, just those alone, starving and poisoning the tissue culture, causes the tissue to break down in its constituent parts, which can contain things which we now know are exosomes, which are way tissues detoxify themselves. But anyways, let's forget about that particular criticism of the whole phenomena of proving tissue culture. Let's see what they found. What they found was they tested their unpurified liquid that supposedly contains virus on 3 different types of human tissue: human lung cancer cells, normal human liver cells, and human embryonic kidney cells. And they said they inoculated them at high multiplicity, which is a technical way of saying they put a lot of stuff on there, right? They put a lot of virus in there. And then they looked at them 24 hours later, and no CPE was observed in any of these cell lines. In other words, the only conclusion that any sane or rational person could come to is that the very way that virologists prove that this fluid containing virus infects and causes damage to human cells or human tissues, what they found was it doesn't. And they make no comment on that except to tell other virologists that, by the way, if you want to grow more virus, don't bother using human cell lines because the virus doesn't grow in human tissue or cell lines. And when you read that, again, the whole thing is sort of shocking because if you just spent using your own technology, using your own methodology, you just actually proved that fluid containing this virus does not affect human tissue. In fact, they said it only affects vero cells, which are monkey kidney cells, which are sensitive to amphotericin and gentamicin. And that's the only way they could prove infection. The final one is shocking for a different reason. See if I can find it here. Hang on a minute. Yeah, sorry. So this was a paper published in the Journal of Clinical Microbiology. So probably one of the most important microbiology journals in the world. I can't pronounce the Chinese guy's name, Yu Zhang something, W. And others, the paper is called RNase resistant virus-like particles containing long chimeric RNA sequences produced by two plasmid co-expression system. That's a mouthful. Journal of Clinical Microbiology. So the point of this study was they were looking at novel ways of characterizing viruses. And so I'm going to read the quote from the study. This is under figure 3 of the study. And again, it's going to sound like gobbledygook, and I'll try to translate it as best I can in a minute. And here's a quote. Lane 1, positive control of PET MS2-3V plasmid using the primers S SARS-1 and HA300RT-A. Lane 2, RT-PCR of SARS-CoV-1 plus SARS-CoV-2 plus SARS-CoV-3 plus HCV plus HA300. Now, what does that mean? What they're saying is they developed new techniques, maybe even a little different, maybe even a little better than the usual PCR. So they were innovative virologists. And what they found was they could characterize different viruses using different primers. These are sequences. And so they were reporting on their characterization of the different SARS viruses. And so they were characterizing SARS-CoV-1, SARS-CoV-2, and SARS-CoV-3. Now, many of us, including myself, actually didn't know there was a SARS-CoV-3. In fact, we just have information that the SARS-CoV-2 was recently discovered because it had never existed before 2019. And there is no report that I know of that there's now a new SARS-CoV virus called SARS-CoV-3. But yet they're using their novel two plasma co-expression system, that's what the title says, to characterize this SARS-CoV-3 virus and actually put a picture of it. And one can only ask, what in the heck are they talking about? Because nobody, as far as I know, thought there is a SARS-CoV-3 virus. But here's the really shocking part of this paper. I could say, anybody want to guess when this paper was published? My guess is most people would say last week, last month, maybe at the end of 2019. The reference is Journal of Clinical Microbiology, 2007. Wait a minute. So 2007, they were characterizing with this method SARS-CoV-2 and SARS-CoV-3. I mean, supposedly it wasn't invented. Sorry, I didn't mean invented. I mean, imagined. Sorry, not imagined. I mean, it didn't mutate into this virus until 2019. So what are they talking about here? And frankly, I don't know what they're talking about. And I don't know how this came about, but it sounds pretty weird to me. So if anybody has any more information, I'd love to hear it. And again, these are, a lot of people will send, oh, here's a paper and here's a picture of this isolated virus. Every single time when you look at it, they found a piece of a hoof and they made a new picture of a unicorn every single time. So don't be fooled by that. These are the actual state of the research on this, and hopefully everybody's got it. And then I just wanted to finish just with a short way that maybe to help people just think about how you can go about imagining or conceptualizing how to keep yourself healthy. And one of the things that I sort of try to conceptualize this and make it clear to people is, as you know, I think that mostly we get sick because of either injuries, like we fall and break our leg. That's different. But the main ones, just like these viral cultures, are we get starved or poisoned. I've also added a fourth one, which I would call, you could also get sick because you're delusional or you believe in things that aren't true. That's also not good for you. But let's stick with the starved and poisoned. So obviously everybody should be eating a nourishing traditions diet like we describe in the book, and that should be very clear. But it turns out we have essentially a 5-step process for keeping ourselves healthy. And what's important about this is if you think like this, you can actually help yourself in many situations. So let's say we're fine, and then we are exposed to some sort of new poison or new stressor or something. And the first way we get rid of any poison is we conjugate it, which means we do a process in our liver to make it soluble, and then we excrete it through the bile, which then gets excreted through bowel movements. So the first and primary way we get rid of any toxic insult is by increasing bowel movements and increasing liver activity, which is part of the whole conjugation. It's like bagging garbage and taking it out to the curb. The point of that is one never wants to be constipated, and one never wants to not have bowel movements every day. And if you do, you should make a concerted effort to not have that happen. And if you do feel like there's an increased toxic exposure, there should be some attempt to increase your liver activity and even increase bowel movements. And this is, of course, nothing new. This is the whole reason for people taking bitter herbs. Those stimulate liver function and increase bowel movements. This is the whole reason for coffee enemas. This is basic natural medicine. If you've been exposed to anything, your body will naturally increase bowel movements, diarrhea, and that helps you flush out the poison, and you can work with your body to do the same thing. There's lots of different techniques of doing that. I don't want to get into them now. You know, everything coffee enemas and Swedish bitters and milk of magnesia maybe, or Epsom salts or gastrofiber. There's lots of different ways of doing that. Now, if that isn't enough, then these toxins can end up in your bloodstream, which you don't really want. You'd rather they only get into your liver, and then the liver gets rid of them through the bowels. But if that isn't enough because you're constipated or not paying attention, then the next step is you excrete the toxins through the urine via the kidneys. And as long as that's happening, so if you're exposed to something, you increase bowel movements, you increase urine flow, and this is usually done by drinking a different variety of pure water and lots of it. So we could use Ophora water or Mountain Valley spring water or whatever good spring or Mount Shasta good spring water you have. But if you're feeling like you're getting sick, the next step is to increase your urine flow, and that's primarily through increasing your water consumption. Now, if there's still a problem, the next mechanism your body uses is to excrete toxins through sweating. And again, this squares perfectly with what we know with natural medicine, because if you've gotten heavy metal exposure or DDT or any other exposure to toxins which are making you sick, one of the time-honored ways of dealing with it is saunas and sweat lodges, and even your body producing a fever, which is another way of sweating out toxins. And one of my points here is as long as you're doing those, as long as you're taking care of yourself through those three mechanisms, usually you don't end up with the disease, because those are the three sort of normal detoxification mechanisms. And we should all be paying attention to those. We should all be paying attention, not only just not poisoning ourselves, but to doing things to increase our liver activity, helping our bowels work regularly, increasing water, therefore urine flow, and doing regular saunas, sweating, or some other physical activity, sweat lodges, some mechanism for getting you to sweat on a regular basis. Now, if those three are not enough, then you end up essentially with toxins in your bloodstream. And the next step your body does is to make white blood cells, which are essentially the garbage collectors in your bloodstream. And that goes along with what the doctors erroneously call bacterial infections. So in that situation, essentially what's happening is there's enough of a toxic load that you start having to generate bacteria to essentially biodegrade and get rid of the toxins. And one of the hallmarks of that is, along with that, your body produces these white blood cells, which are like sending out the garbage crew. And in that case, you're actually sick. So then you recruit the fever and other chemicals to help urine flow and to make you sweat. And now you're actually sick. So you don't want to get to that stage, but that is the stage when the body starts making white blood cells. And that's the stage when you start seeing so-called bacterial infections, which is really nature's way of helping you biodegrade the toxins. And the fifth stage, if all those are still not enough, is then you start making antibodies. Now, we are told that antibodies basically help you get over viral infections, but as I pointed out a number of times, they don't really mean that. Antibodies are just the fifth stage of you've got enough toxins in your bloodstream and in your tissues that your body is forced into making these neutralizing chemicals called antibodies, which actually attach to the toxins so that you can recruit the garbage collectors and maybe even get rid of them through your kidneys and urine and sweating. And once you're in a situation where you're always making antibodies because the other four steps are essentially filled up, so to speak, now you're talking about somebody with an autoimmune disease because they are in a chronic state of excessive antibodies. So we don't want to get to that stage. We don't want to get to the stage of having hyperinflammatory responses based on elevated white blood cells. We want to stay with the first three. Increase your bowel movements. Use bitters to help your liver. Increase water consumption. Sweat. Go for walks. Ground to the earth. Expose yourself to the sunlight. All these things keep you in the first three phases so that you don't end up getting what we erroneously call being sick. So that's a very simplistic way of looking at it, but sometimes simple things help people actually navigate how they can take care of themselves and their family with a simple model that tells you what you should be focusing on. So with that, I want to thank everybody for listening. I hope you keep track of our website at Dr. Tom Cowan. I hope you order not just one or two, but many contagion myth books, spread them around, and just keep in touch, send me comments, send me questions, watch the Weston Price thing we're going to be recording today. And again, I'm grateful for all the support, and everyone take care.